These are the solutions for manual prep. Solution 1 contains 50 millimolar glucose, trichloride, RNase A and EDTA. This is solution 2, lysis solution, and this is solution 3, neutralization solution. So among all three, solution 1 will be kept at 4 degree because it contains RNase A. The pellet of overnight grown culture from which we are isolating plasmid by manual prep. For that, this is 7.5 ml culture, so we are adding solution 1, solution 2 and 3 accordingly. I am going to add 350 microliter 300 microliter solution 1, I have transferred all the content in the centrifuge tube. Now I will add 300 microliter solution 2, by inverting several times. It has become little bit clear than previous one. So now we can add solution 3, and then we will split it down. 400 microliter solution 3. It will precipitate the cell debris and look like this. I'm doing centrifuge at maximum speed, which is 14,000 rpm for 10 minutes at room temperature. So it's done. I'm transferring the supernatant in fresh tube, and this is cell debris pellet. add same volume of isopropanol, and we'll keep it at -20 for half an hour. This is isopropanol. After half an hour incubation at -20, we are again centrifuging the solution at 4000 rpm for 5 minutes, 14,000 rpm for 5 minutes. So this is the DNA pellet, plasmid DNA pellet, and we have to remove all supernatant, and we'll wash this pellet with 70% ethanol. I am adding 600 microliters, 70% ethanol. Now this is plasmid DNA pellet, and whole 70% ethanol has been evaporated. So now I am going to add 200 microliter RNase containing milletube, and the RNase treatment is given so that any contamination of RNA will be terminated, to avoid RNA contamination in our plasma. Now I am resuspending the pellet. After resuspending, I will keep it at 37 degree temperature for half an hour.